I will present NeCEN, which is the Netherlands Center for Electron Nanoscopy. And this is a nice picture of Leiden, of the harbor of Leiden. If you haven't been to Leiden, this is a very pretty city south of Amsterdam. So if you don't know, the Netherlands is located here in Europe, just north of Belgium and France. And Leiden is in between The Hague and Amsterdam. It's very close to the airport. It's a 20 minutes train ride. So it's easily accessible, and we're close to the beach, very close to Amsterdam. This is a typical slide from the Netherlands. You might think Netherlands is tulips, windmills, and paintings. This is what Dutch are known for mostly, and they're also very good in water controls and canals in the cities. This is a picture of Leiden at night, which is a pretty city. Last 2 weeks, for the first time for me, I've been in the Netherlands for 5 years, but for the first time I've seen snow and frozen canals. So I managed to go skates on the canals in Leiden. This was quite extraordinary. And on the right here is a nice, beautiful picture of typical Dutch landscapes. But as I said, this happened just once for me, and most Dutch people said it hadn't happened in the last 10 years. So that was quite something to try. All right, let's move on to the science a little bit. So I've split the talk in 2 parts. The first part will be presenting NeCEN, who we are, what we do, and how do we operate. And the second part will be an example of sample to structure service, so something that we can offer through iNEXT-Discovery. As I remind you, iNEXT-Discovery aims to enable access to structural biology, and mostly for non-experts in structural biology. So that's something we can really assist non-experts in doing. Let's start with the first part. So who we are. NeCEN is owned and is part of Leiden University. So the governance, NeCEN is owned by the science faculty and the Leiden Medical Center, both part of Leiden University. It started in 2011 as one of the first open access national facility for cryo-EM, equipped with 2 Titan crosses. We operate with a synchrotron model. That means that anyone in the world can apply to NeCEN. We have users from Korea, India, Canada, anywhere in the world. This is a picture of the science faculty buildings. This small, can you see my pointer? Maybe I'll make well, anyway, this was this is the old place where NeCEN was in 2011 to 2016. This was temporary building because the faculty knew they were going to move to a new building, which is this one. So now we moved in 2017 to this new part of the building, and this building highlighted in red and blue, they are both demolished, and work has started to continue this building to connect to the circular one here. So in the end, hopefully, in 2023 or a bit later, maybe now due to Corona, we'll have a full building connected with all these beautiful pipes. And it's truly a beautiful building. It's really nice to work into. So at NeCEN, we have all necessary equipment to support your cryo projects. Maybe something to note is that we are a BSL-2 laboratory, so we can handle BSL-2 samples. We can help you with freezing the sample, we can store it, and we have our own storage system that we've designed, so we don't have these commercial pucks, but we have a home-made pucks, and all the drawings for these pucks are available in this publication. So that you can fit 15 grid boxes, I believe, in each puck. And because they are homemade, you can design it the way you want, basically, and the size can be adjusted. So we have clipping tools, cartridges, and all the classical equipment for thermometer microscopes. So at NeCEN, we have 4 electron microscopes, 2 Titan crosses, which are the flagship instrumentations. All these machines are in BSL-2 except Titan 2, but it will be in BSL-2 very soon. Titan 2 has a K2 bioquantum and a Falcon 3 camera, and it has a CS corrector. Titan 1, which is the preferred machine for most users for single particle and tomography, has a K3 bioquantum and can collect up to 400 images per hour with AFIS data collection. Very recently, we had a user collecting 5 hours of data and getting a sub-3 angstrom structure in the next 2 days after data collection. So this is really a tremendous speed. You can really achieve multiple structures in 1 day now. We can also train users on those courses. And then next to those two flagship instrumentation, we have a smaller TEM for helping users to screen their samples in stain or cryo. This is mostly for local groups. And we also have an Akilos cryofib to prepare lamella for doing electron tomography on thicker samples. And this is not yet a service that we offer. It's mostly a research instrument, but we are planning to open it as a service in the next few weeks. So in terms of services, as I said before, any user, any scientist can apply, anyone in the world. It can be academic or companies can apply to NeCEN. And we have different types of services. We don't do biochemistry. That has to be done at your home lab, but within iNEXT, you can get support for that as well. Or also in Leiden, we have access to a protein facility for supporting biochemistry experiments. At NeCEN, we can do NeXT-STAIN, which we mostly do as a service. We do data collection on the crosses. We do 3D reconstruction also for companies or as a collaboration with academia. So to access NeCEN, there's mostly 2 modes: open access and reviewed access. So open access, that means you can go directly on the NeCEN website and there's a button that says access application. And after you create an account, you can just send us a form to apply for beam time at NeCEN. And the application is reviewed only by the NeCEN staff, so it's private. And we review mostly on technical feasibility, not really the high scientific quality. So, for example, for companies, it's quite attractive, or for people that are in a rush to publish or have a very competitive project, that is really attractive. At the moment, the price for 2021 for international users is $22.50 per 24-hour beam time. And as I said before, you can collect 2 or 3 data sets in 24 hours. And then the second mode of access is reviewed access. So we are part of different consortia for this reviewed access. I will just have 1 slide per consortia. So the first one is the NEMI, which is the Netherlands Electron Microscopy Infrastructure. So NeCEN is just 1 node within this electron microscopy infrastructure, but we are unique in this infrastructure, offering 300 kV access to Titan crosses. And you'll see other nodes have different technical expertise. There's also the Instruct ERIC, obviously. We are part of a group of 11 EM service providers spread all over Europe. And of course, you know about the iNEXT-Discovery. We're part of all of these 26 partners, and 9 of them offer EM service. So in the reviewed access, in those 3 consortia, the applications are reviewed by externally, by scientific staff externally. For Instruct ERIC, there's a few points that researchers need to be a member of Instruct member countries, and the price of the data collection is highly subsidized. There's no limitation on the time that you can collect data, but you have subsidized access, and then you pay the difference. For iNEXT-Discovery, everything is free. As long as your application is reviewed positively, you can come to NeCEN, and the shipping of the sample is supported, data collection is supported, and we can also support screening of sample. We can only deliver a certain fixed number of days per year, but apart from that, there is no limitation. And it's a bit similar for the NEMI. We have a limited number of days per year that we can provide. The price is highly subsidized, but it's restricted to Dutch researchers. But it's good to say that any researcher in the Netherlands can apply through NEMI to access NeCEN. We also offer training and education. For example, we have developed cryo-EM schools together with the Offshore Scientific. We started to provide those schools in 2017, I believe it was the first one. And then from those schools, you might have seen now these online lectures. So those lectures derived from the first EM school. We trained operators of the Titan cryos. And this is a commercial course, sorry. We also offer one-on-one training. It can be a one-week training where we train you on one-on-one on sample preparation and everything you need to know to operate cryos. And then we can also have more individual training or tailor-made training, sorry. So in terms of data collection, so as I said, we have GATAN K2 and GATAN K3 detectors. We provide access for 65% of the time to the Dutch groups through NEMI, and 25% to European researchers or international researchers. And for approximately 10% more or less, it's industry access. And it's almost 50-50 single particle and tomography data that we collect. And all the single particle is collected with EPU, while most of the tomography data is collected with StereoLEM. And this the tomography has increased in 2019, and that is due to the NEMI project that is really started in 2019, because a lot of Dutch group work on using tomography. And it's been stable in 2020. In terms of data quantity, we have 700 terabyte local storage, because we produce something like 600 terabytes of data per year. And now with the K3, this is only increasing. So we all know about Corona, of course, and I've listed here all these notes from the University of Corona policy that keep on changing. And I see now I've stopped after 15 October, I stopped keeping track. But because of Corona, we had to adapt the way we work. Obviously, we couldn't have as many people at the facility as before. So we've developed or we've implemented more remote data collection and remote monitoring options for users and for our staff. I'll just present the two main options we use. So the first one is a simple TeamViewer that we connect to the camera PC or the support PC, and that allows user or the staff to control StereoLEM directly on the camera PC or to set up EPU that is on the Cryos PC. So here you have a window with the Cryos PC that is viewed from the camera PC. So it's just a simple VNC connection. And what's good about it is that the screen can be locked and you can prevent the users from clicking anything. It's just a view access. So it's really good for remote users to help us or guide us where to select the right areas to collect data. And the second option is a VPN. So we have a VPN server that is managed by the university. So this is a bit more cumbersome because you need accounts, and those accounts are managed by the university. But both our cryos are under this VPN server. So that allows us to develop a software together with FEI, being able to connect remote pads for remote sites. So someone in Utrecht, for example, or Amsterdam could have this remote pad connected to a computer and could control the cryos directly through the internet, as if he or she were in front of the cryos. So that we don't really allow anyone from outside the Netherlands, but we've tested it with 2 sites in the Netherlands because in this Corona time, people couldn't really travel anymore. So that seems to work very well. Sometimes the speed is a bit low, but it helps users for operating remotely. And of course, we've also developed monitoring of data collection to help users make the right decisions. so as most labs have nowadays. The first option that we provide is warp. So this is the screen on the right here. So it's a simple warp software that is processing the data, and then data is directly uploaded for the user to download directly while data is being collected. And the second option is the homemade pipeline, which is a bit more tailor-made to the operator, also showing all the necessary information for the operator to make decisions during data collection. At the end of data collection, we provide a nice PDF report showing some of these values to ensure the quality of the data set was optimal. And then, of course, when we do this, we upload data to a remote portal. So as soon as you start data collection, the data is uploaded online to a company that we use based in Amsterdam. It's called Surfsara. And accounts are managed by the company, and everything is private. So as soon as data collection starts, data goes there together with the pre-processing data also goes there. We share the data with the user, and they can right away download it on their own server. So to continue processing if they wish already, and get direct feedback during data acquisition. Also, all this data that we generate, we try to organize it. As you may know, when you collect a movie, there's a lot of metadata collected with it. So this metadata, it would be good to use it. So that's what we try to do. So we have organized all our projects into a website, and behind this website, there's a database with all the metadata that is kept. And we try to keep information on the speed, on the grid type, anything we can extract basically from the metadata itself, but also from the user, what they can tell us about their project. And then we also have developed a wiki to help users with protocols and explain as much as we can about the structure of the NeCEN network and so on, so that when people are remotely accessing the cryos, if they have a question, they can directly go on the wiki and check if there's an answer for that already. And on the left side, we have a management software, which is called PPM Stratcore, to manage all the projects and all the users. And now we try to link all of these together into one system. So it's not there yet, but that's something we try to do, so that everything will be into one umbrella and all the projects will be into one umbrella. So now, because of the K3 mostly and the remote work, the day-to-day work has changed a little bit. I mean, before we would do one sample per day, and then we would leave and still collect the same sample overnight, but this has changed. So that's another reason to really promote remote access, because in the morning we still load 4 to 8 grids, but by noon or just after noon, we hand over the session to the user or to the operator, and then they can collect data. Or even before noon, we would collect a small data set, and then at 3 p.m. we would collect a second data set, and then at 9 p.m. if the user can remotely connect, he will start another session again overnight, and so. And on and on. So you could collect 4 or 5 data sets in 24 hours. And I know ThermoFisher is now providing a EPU version that will do this automatically, but that's not something we have at the moment. And if you leave sample with us, we'll ship them back the next day, of course, and then we'll send you all the report that we can, and we will ask you for feedback during the same period. Feedback is quite important so we can improve our services. So all this remote access work that we've been developing allowed us to provide a continuous service during the pandemic last year. We've supported users that worked on Corona. So this is an example of a coronavirus study on the replication of coronavirus. which is a drug target, obviously. So the users used our Achelos and then our Titan Cryos, both with K2 and K3. And this was published middle of last year. And then the second example would be the sample to structure service that we have done also last year during the pandemic. And just to be clear again, this is not an iNext project, but that is something that an iNext project could look like, basically. So the work I just want to present for the next 5 minutes is a collaboration with a company, Janssen Vaccine and Prevention, which is part of Johnson & Johnson. So they came to us last May, and the goal of the project was to generate a closed spike of the SARS-CoV-2 trimer for vaccine development. So as you may know by now, the S trimer is the sole target for neutralizing antibodies and also the main focus for vaccine development. But that is something that is metastable. So having the ability to generate a closed trimer was essential to develop, to improve vaccines efficacy. So Janssen was developing different constructs, and we supported them by screening some of them. So we started with, enhanced support, if you wish. They would give us the tubes of the different constructs, and we would screen them by NeXT-Stain first, using the Talos. So I believe we screened 7 different constructs. It took approximately 10 days of data collection and continuous processing. So this is example 1 where we generated some 2D classes and 3D classes as well. And then the second one, you look, the 2D classes, they looked a bit better if you compare to this one. Some of them are not so nice and so on, but the 3D looked quite okay. And the 2D classes here, they looked much better, but the 3D was not great. And then the third one was the better one, even though the micrograph is not ideal, but the 2D classes and the 3D classes were beautiful. But this is NeXT-Stain. It's really hard to say much about 3D classes in NeXT-Stain. I never really trust it. So of course, EM is not the sole technique to really judge your constructs. And there was a lot of biophysical characterization done by Janssen on the side at the same time we were doing the negative staining study. There are trimers expression levels, they were checking with binding, they were checking temperature stability and all of these techniques. And of course, within iNeXT, you can also do this by accessing the biophysical facility at the NKI, which is part of iNeXT. So that's also something that can be done within the iNeXT project. And then once we chose a couple of constructs to go for cryo, we moved to the cryosys. We collected 2 of them, as I said, and it was quite straightforward, to be honest. We froze 8 grids in total, and we collected 1 day for one of them, but it was not so great, so we moved quickly to the other one. And in total, we collected 3 days at the cryos. Almost 10,000 movies were collected in super resolution. And these are the conditions we used. And this is a typical micrograph. And during data collection, we set up some reliance schedulers and some loop of processing. And we obtained fairly quickly nice 2D classes on the fly during data collection. So after that, we continued with the processing, and this took a bit longer because there was a big data set. So this took 3 weeks to really go to the end of the image processing. And of course, here is a simplified workflow for the image processing. There's a few iterations at each step, and sometimes you go backwards and try again. So from this 10,000 movies, we selected 1.4 million particles, and then out of this, almost 900,000 went to 3D. As you see, the 3D model, which is an ab initio 3D model, it doesn't look very great. But as soon as you do 3D classification, you can select the best subset, which is 800,000 particles. So not so many were really bad, but the bad ones really made this look ugly. And then we continued to to isolate a closed and an open conformation. But it is to be noted that the closed conformation is approximately 2/3 of the well-formed particles, while the open conformation is 1/3. And resolution was below 3 angstrom in both cases, so we could build an atomic model, which we did together with Janssen scientists. And they are both deposit at the PDV if you want to check. And these are the final statistics for the image processing. And of course, as I said, this is something we can do with Janssen, with iNEXT-Discovery, sorry. We can support you for sample preparation, screening, image processing, model building if needed, not just data collection. And I just have a last slide with different examples of other studies we've done in the last 2-3 years, where we do simple tomography of on liposomes, or we study some BSL-2 virus with a group in Spain, or some DNA binding proteins with, I think, enhanced support as well in there. And this one, the user collected himself, so the data himself. So there's a range of user expertise that we can support. And finally, just to acknowledge the funders. So NeCEN is owned and funded mostly by the University of Leiden and the Leiden Medical Center, and the NEMI, which is a project funded by the NVO, which is the Dutch Science Foundation. And of course, I would like to thank Instruct Eric, iNEXT-Discovery, and Instruct Ultra project that funded the remote access project. And I would like to thank you for your attention.